to be here.
We will do that.
Alrighty. Hello, everybody. Welcome to this feature research session that we are going to be discussing studying tissue samples from rare cohorts to learn about early stage AD pathophysiology. I am Ollane Uriarte Duarte, the director of the Strategic Fund at the Alzheimer's Association, and I'm here with my co-chair, Malu Tansi.
Hi, everybody. Thank you for coming. Malu Tansi, professor of neurology at Indiana University School of Medicine. It's an honor and a privilege to be here, and I think you're in for a treat because the findings from this panel have been pretty transformative, I would say.
Good. So this session will highlight collaborative research using the IMPH cohort to deeply interrogate the underlying pathophysiology at the early stages of ID. I believe we have an amazing set of speakers that are going to be sharing their insights from different perspectives using the same cohort. And I'm looking forward to hear their presentations and then all the questions and discussions at the end. Just a reminder that we are going to hold questions until the end. So for those that they are here in the room, please approach all the microphones and we will get in order all those questions. And then the ones that they are joining us virtual, don't forget to submit your questions, indicate which is the person that you are directing your question, and we will make sure that we read those. So without further ado, it's my pleasure to introduce our first speaker, Dr. Leinonen. I knew I was going to pronounce this wrong, sorry. He's going to talk about normal pressure hydrocephalus, potentially treatable dementia, and window for living human brain.
Okay, thanks for the kind introduction and overview introducing the invitation to come here. I first say a few words about normal pressure hydrocephalus and then go further why this disease is relevant for the Alzheimer's disease research as well. If you want to know more about NPH, there is tomorrow actually a session for comorbidities where Dr. David Jones has another talk on NPH. I have nothing to disclose. I start with my acknowledgements. I want to acknowledge my research group, especially Anssi Lipponen, the data manager. He's here today as well. I had yesterday also a poster on the NPH topic. And also the funding sources, including Alzheimer's Association. Then my most important collaborators here, and we can see today Tarja, Henrik, Evan, and Sam from Bet's lab soon. So NPH, it's up to 5% of all dementia. So it may not be too rare, and I think every neurologist who is working with in the memory clinic should be aware of this disease. It's potentially treatable by CSF diversion. If the main symptom is gait, it may be NPH. If the main symptom is cognition, it may be something else. But if you have, at the same time, gait problem, cognitive problem, and even urinary incontinence, you have to consider this. And if you have a brain imaging, typical signs of enlarged brain ventricles, and differed from typical atrophy disease, where you see cortical atrophy, in NPH you usually have the obliterated high convexity sulci. We already know that there are some potential risk factors in genetic part, so there is potentially a familial form of NPH. And then we know the secondary NPH, any severe brain insult like TBI or subarachnoid hemorrhage may cause a secondary NPH. But what this cohort mainly is the so-called idiopathic form. But if it's idiopathic, we just don't know the reason. We already know that there's a genetic risk and then there must be some kind of environmental factors that we just don't know yet. We know that years before the clinical symptoms, we may see enlarged brain ventricles. It's called AVIM, so it's like an asymptomatic ventricular enlargement. And then finally, if you just live long enough, you may get the clinical disease when you have the case for the shunt. And again, you may have another risk factors for the other diseases, like Alzheimer's disease. So if you are ApoE4 carrier and have NPH, you are in the same risk of having AD than anyone else. So the final outcome of these patients is actually determined by the potential comorbid neurodegenerative diseases. So shortly about the clinics. So if the gait symptom is the only thing, and you may have like reflex changes, you have to consider like spine problems. It's usually typically like instability, magnetic, broad-based, short step length and slow turn. If the cognition is the only symptom, and especially or even the primary symptoms is often something else. The cognitive profile overlaps all the other memory diseases. We don't have a very specific NPH-type cognition, but typically they are like psychomotor slowing or like impaired executive functions. And the urinary incontinence you can find from nearly everybody if you ask if they have an urge. And finally, when the disease goes on, you even may get the fecal incontinence. If the patients have rigidity or tremor, it's usually something else. From the anatomy, of course, we have to have the enlarged ventricles. Typically, we have a narrow callosal angle compared with atrophy, where you have a very wide callosal angle. And the parasagittal sulci may not always be very obliterated, but that's considered the typical finding. If you have an enlarged sylvian fissures, you have to keep in mind that it may be very typical for NPH, not just considered as atrophy. And the same is with the hippocampus. The temporal horns may be enlarged because of the hydrocephalus, not just because of the atrophy. And sometimes after shunt, you actually increase the volume of hippocampus because it looks bigger and it's bigger. There are a number of anatomical subtypes, which means that it's not the same disease for everybody. In the glymphatic type imaging, so it doesn't matter whether it's gadobutyl or whether it's like radioisotope, you see a reflux in the ventricular system and you have a stagnation that the agent is not cleared so rapidly as it should be in the normal subjects. We know that it's progressive disease. If you delay the treatment, you lose the brain, and the lost brain is lost forever. So you have to aim to early diagnosis. If we treat them properly, and they don't have any other severe brain diseases, the prognosis is actually close to the age-matched population. So it's really worth of making the diagnosis, really worth of treating them. You may get very happy patients. So about 1 quarter remain cognitively normal if they are treated. 1 quarter get at least mild cognitive impairment, which might be very stable. But another half goes for dementia if they just live long enough. And in that case, it's usually something else than NPH. It's like Alzheimer's disease or vascular type dementia, or then anything else. So that's why in Kuopio, we ask the neurologist to see the patients also after the shunt and not just before. There's still debate in the neurological society if there is NPH at all. I believe since we, for example, like in Sweden, they have found even identical twins without any other brain diseases. And I think this is the perfect disease model both for the genotype and phenotype. Like any other neurological problems, you should ask the family history, and not just whether they get shunt or NPH. You ask what kind of symptoms they had in the previous generation, because the story may be very similar. They just have not treated ever. We know in the pediatric hydrocephalus, there's a large number of candidate genes, even disease-causing mutations. Many of them are linked with other CNS abnormalities. And it seems that in the early onset hydrocephalus, the cilia abnormalities play often a key role. But when we go to chronic hydrocephalus, this NPH in the elderly population, it seems that we can't replicate any of the cilia genes in this late onset disease. So it's a different disease. In the FinGen, which was actually the first ever genome-wide association study in NPH with 1,500 cases, we found actually 4 loci that were significantly associated even in the INPH cohort, which was like cleaned from those with the secondary cases. Only one loci overlaps other neurodegenerative diseases. It's actually the top hit, which is related to PSP as well. This is very interesting, since the brain imaging of PSP and NPH may look very similar. It may be it's just the clinical, it may be just coincidence and overlapping of these diseases. And the other hits are actually already associated with brain ventricle volume, and some of them even with the CSF-AD biomarkers. The top hit, especially, it's related with the organic anion transport, for example, and it goes very well with the hypothesis that the NPH may be a glymphatic disorder if there is a glymphatic system. It just depends what term you want to use. But in the end, it seems that the NPH is more like a clearance problem. And this is our current hypothesis for further research. So now we go to brain biopsy. During shunt surgery, we have this possibility that we have a place where we put the shunt through frontal cortex. We can have the tiny biopsy, which is actually the same size than the catheter we put inside of the brain. We can have a cylinder biopsy where we see the fresh frozen samples that are analyzed by Evan's lab. And then nowadays we take a small pyramid, get the samples that are analyzed by Tarja, who will soon show her results. So we make a diagnostic stain. If we have an amyloid pathology and hyperphosphorylated tau in brain, it means actually a definite neuropathological Alzheimer's disease biologically. If you have only amyloid, it's like 10 times risk of getting Alzheimer's disease in the future compared with those with totally normal biopsy. This is what we have seen during the last over 10 years. For some reason, the red is increasing, so those with both amyloid and tau. It's for some reason they are increasing. And then the blue is comparing those with early AD, who just have the amyloid only. And then we see very few TDP for the 3 cases as well. We see that the biopsy affects the cognition, so it has a prognostic value. If you have normal biopsy, you often can even improve by cognition. That is the part of cognition that is decreased by the NPH. And then if you have amyloid and tau, it seems actually quite bad. But even if it's only amyloid, you see it not so well. Around 1/4 eventually have Alzheimer's disease, for some reason a little bit more frequent than the general population. And the amount of amyloid in the biopsy correlates with the outcome, so it matters. We see rarely other diseases than Alzheimer's disease as well in the biopsy. For the CSF biomarkers, for some reason, they are lowered in general. But if you use the A-beta phosphatau ratio, it should be pretty okay. But don't exclude the patients. We see nice correlation with the biopsy and the plasma phosphatau 217. And actually, to conclude, the brain biopsy pathology in possible probable NPH predicts the cognitive decline and comorbid Alzheimer's disease. It correlates well with the CSF and plasma biomarkers. It correlates with the amyloid PET. It correlates with the postmortem. So the brain biopsies in these surgeries could be useful for the AD, to study the AD pathological process. This is just the biobanking we do for these patients. And it was nice that 2 years ago this was featured in ALZ Forum, the whole process, and you still can find them. And I want to end here to take home message that don't forget this diagnosis. Up to 5% in dementia clinic may have this disease. It's progressive, but you have to keep in mind you may treat this. It's still the shunt is the only effective treatment. And neurologists should see the patients even before neurosurgeon, because the neurosurgeons put every patient get the shunt or no one get the shunt. And sometimes the shunt valve adjustment may be useful in the future. And this gives us actually a window to take also the follow-up CSF. And the comorbidities actually determine the final outcome. Okay, and I think next you will see what this window for the living human brain means. Thanks.
Thank you very much, Ville. So now our next speaker is Dr. Makosko, and he will explore cell states associated with amylopathology and cognitive resilience.
Thanks very much. It's great to be here, and especially great to be sharing the stage with so many valued collaborators and colleagues. I'm going to be telling you about how we've used our single-cell analysis approach to, in combination with this amazing fresh biopsy cohort, to start to learn about cell states and neuronal vulnerabilities that are associated with amyloid pathology and also more recently cognitive resilience. So I think Alzheimer's disease has two major sort of big picture challenges. The first is that the progression of the disease is quite slow and insidious, and by the time you start to see clinical manifestations of the disease, there's a lot of biology that's been brewing for potentially many years. And of course, we're very eager to understand what that biology is, but the challenge is that where the biology is occurring is in a piece of tissue that is unfortunately only accessible at the time of death. And so if you really want to understand, for example, how somebody, what kind of cell states or vulnerabilities occur at the beginning of the disease and how those different cell states may associate with the progression of the disease, it's very challenging. You only can look back retrospectively in postmortem tissue. And so what we really are excited about with this cohort is the ability to track living patients and see how their cognition changes over time, potentially how their biomarkers change over time, and that can really allow us to really make credible associations since we now have these brain biopsies that we can then examine and analyze prospectively. So Villa already, of course, introduced this Quopio cohort, but from our perspective, what's exciting is that they've drawn these biopsies from many hundreds of patients. We've been able to sample 295 of them by single nucleus RNA-seq. And they've scored all of these for histopathological burden. And of course, we also have longitudinal cognitive data on these patients, and also paired CSF, blood, and whole genome sequencing, which really allows us to do these kind of multimodal analyses across different fluid compartments. And you'll hear from Tarja soon about her own analyses using acute slice physiology to study the actual physiological behavior of the cells in these samples. And so it's really an opportunity to link AD pathology, cognition, and biomarkers in living human brain specimens with actual cell states that we can sample by cutting-edge genomics. And really, what I'll emphasize here at the end is this really incredible opportunity also to track patients prospectively after biopsy, which is quite unusual. I'll just mention a couple insights that we had from our initial study of these samples a couple years ago that we published, where we looked at 58 of these specimens, and we had sort of three key insights, and a couple of them will appear later in the other work that I'll present. So the first is that we found there was a selective loss of certain neuron populations at the very initial accumulation of amyloid. And one of them that was most prominent, shown on the top there, is this layer 1 interneuron population marked by a LAMP5 and NDNF expression. But these are upper layer, layer 1 exclusive interneurons. And we found that if you had lower amounts of these layer 1 interneurons, you tended to also have increase in expression of activity-related genes in your layer 2-3 excitatory cells. So that's shown on that plot on the dot plot on the left, where the layer 2-3 excitatory neurons are showing more like FOS and Jun-B expression. And then Tarja has been able to start to validate, this is the beginning of this work that you'll hear more about, to show that in donors that have amyloid beta accumulation, they are more prone to these like long trains of excitability in response to NMDA. The second thing we found was that both excitatory neurons and oligodendrocytes produce amyloid. This was just initially a hypothesis that was generated from the gene expression data itself. We found pro-amyloidogenic genes expressed in both excitatory neurons and oligodendrocytes. And when we went and made iPS cells from both of those different cell types, we found that they both produced on a per cell level the same amount of amyloid. And the final thing we examined in great detail were microglial states, because we had this opportunity to see microglial states in vivo tissue. And of course, we're interested in microglia in particular because they're loaded with the expression of genes that have been associated by genetics with a risk for Alzheimer's disease. So we made this very detailed and very large integrative analysis of both our data and also data from ROSMAP and other postmortem data sets. And we found that those cells, in response to amyloid, all tend to upregulate or dysregulate, I should say, genes that are included in this GWAS. So there's an enrichment of 80 GWAS genes in the differentially expressed genes amongst these microglia. So now, and in addition to that, what we found is that there are many different cell states that are shifting in response to amyloid. So this is our final data set now that we've generated a total of 295 biopsies. And just to call your attention to the fact that there's substantial increases in expansions in multiple different populations. The first set of populations are sort of what we would typically describe as DAM, disease-activated or disease-associated microglia. These are cells that express genes like GPNB, high levels of ApoE and LPL. But there's also expansion of other populations, including this homeostatic population that caught our attention, HM3. And so one really outstanding question that we've had, and I think many have had in this field, is how do we make sense of the relevance of these particular reactive states? Are these states protective against Alzheimer's disease, or do they sort of connote harmful effects longer term? And so this is analysis that's been spearheaded by a computational biologist in my lab, Vishal Sarsani. And really what made this particular question even possible to answer is this cohort, because every one of these patients has been followed prospectively for their cognition over a median of 2.5 years, but in many cases much longer than that. And so by following these patients over time, we can actually make a model where we ask, given a certain amount of amyloid pathology in these specimens, and a certain cognitive trajectory, and there you're seeing two different sort of summated trajectories of A beta minus, tau minus specimens that have a slight increase actually in cognition trending over time versus those that have amyloid beta that have a slight decrease in cognition over time in this cohort. Given those different cognitive outcomes for each patient, are there certain cell proportions that may moderate or mediate the transition from an amyloid pathology burden to a particular cognitive outcome? And that's what that model is describing there. So now we can effectively annotate these microglial states. We have these states expanded or contracted in response to amyloid, and we can just superimpose on top of that a cognitive cognitive effect onto those particular states. And so what you see there is that the LPL CD83 positive population, which is this sort of dam-like state that has been described, the more you have of that state, the more likely you are to have a cognitive decline, whereas other expanded populations like this HM3 population actually seems to connote some cognitive resilience the more you have of it in the tissue. So what are these states? We're exploring this actively, and there's lots of really interesting biology here, but I just thought I'd mention a couple things. The first thing is that in HM3, you see this very high expression of homeostatic genes, even more so than the sort of the bulk of the homeostatic cells. And they also seem to have high levels of expression of phagocytic and autophagy-related genes. In contrast, the LPL CD83 population, much better described, has this high expression of lipid metabolism and glial cell activation genes. So what Vishal did is he just made a ratio. Since we know that both of these populations are now expanding in response to amyloid, he made a ratio. And he just asked for donors that have a higher than median level of this ratio versus donors that have a lower than median level of this ratio, how do their cognitive performances occur over time, change over time? And what he found was that there was a several-fold, 2 to 3-fold increase in the risk of a substantial drop in their mini-mental status at 5 years if they have a below median ratio of these two microglial types versus a high, above median ratio. And we were able to partially validate this in the ROSMAP cohort because we, of course, can't look at the exact trajectory and predict prospectively in that cohort, but we could see that a higher versus lower ratio of this set of microglia did predict a decline in mini-mental status exam right before death. So last thing I wanted to mention is just this question of like, how are other cell types affected by these proportions? We just looked at 2 different cell types, the HM3 and the LPL, but of course we have access to all the different cell types that are found in these biopsies. And so the first thing we found, which I think was quite encouraging and sort of validating in some way, is that HM3-LPL ratio really associates with a lower astrocytic reactivity. So we have all these different glial populations here. They're sort of named in a rather arcane way, but you can see here that this AST-RT3 population is quite depleted in samples that have a high level of this HM-LPL, whereas a high level of this HA1, this homeostatic astrocyte population, is relatively increased in the high HM3-LPL ratio samples. And it turns out that this RT3 population is loaded with genes that have been identified as sort of A1-like astrocyte genes, initially identified by Shane Little, Ben Barris several years ago. And actually, those A1 astrocytes are also defective in phagocytosis. So just as HM3 seems to be highly phagocytic, it also seems to be decreasing this sort of phagocytosis-impaired population of astrocytes. So lastly, what about neurons? And this is where our previous work started to intersect with this new finding. And that is that when we look at the impact of a change in this ratio, so we're just asking, like, what are the cell types that are most influenced, the neuron types that are most influenced by changes in this HM3-LPL ratio? This is quantified by something called the transcriptome-wide impact score. And what we found is that the cell type that had the highest sort of effect due to this change in ratio was actually the same layer 1 interneuron population that we identified as highly vulnerable to amyloid. And actually all the other top populations here that are in red are all also upper layer or layer 1 exclusive populations. So this is just to remind you of this sort of initial study where we found this selective depletion of these cells in these amyloid-containing specimens. And actually the most sort of impacted layer excitatory population are these layer 2-3 excitatory cells. So we really think that we're now identifying this sort of state that, or a shift in the sort of the overall state of the tissue that seems to lend samples with high levels of amyloid towards a more cognitively resilient state. So in conclusion, we found both amyloid-reactive microglia that are both harmful and protective for cognition over time. The protective microglia are enriched for genes that are associated with autophagy and phagocytosis, whereas the harmful microglia, the DAM microglia, are laden with lipid-associated genes. The ratio of protective to harmful microglia seems to predict cognitive resilience across cohorts, and a high ratio is associated with less astrocytic activity and a strong effect on layer 1 interneurons. So what we're now leveraging this cohort to do going forward is we've been asking, why are these layer 1 interneurons particularly sensitive to these microglial state changes? We'd like to understand where these different microglial states are in the tissue. We're building a biomarker signature of the microglial ratio to sort of see if we can identify donors in other living cohorts that don't have brain biopsies that might have differences in this microglial ratio. And we're performing genetics on the microglial ratio because we now have 1,000 donors in this integrative analysis with all the postmortem samples that can allow us to see genetic influences on this particular ratio. And then, of course, in collaboration with Beth Stevens' lab, we're very eager to start to think about how we can generate and manipulate these populations in vitro. So with that, I'll just acknowledge what a terrific collaboration this has been with all the folks here on the stage, and particularly to recognize Vishal, whose work I mostly presented today.
Thank you, Evan. That was excellent. Our next speaker, Dr. Tarja Malm, is going to present how early AD-related pathology alters neuronal circuits using slice electrophysiology.
Thank you for the introduction. It's a pleasure to be here. I'm really excited about this session and to present you our data. And I would like to, I have nothing to disclose here. And I would like to start this presentation by acknowledging my awesome lab who do excellent work on these valuable biopsies. And of course, all the wonderful collaborators and who make this work possible, as well as the funding agencies. So basically, whenever we get the biopsies into our lab, we slice it up into 315 thick sections, from which one goes to spatial transcriptomics and single-cell sequencing to Evan McCusker and Beth Stevens' lab. Another sample we use for patch clamp studies, and another for multi-edited recordings. And after these recordings, we subject the tissues to in-depth tissue histopathology. We also collect additional slices only for immunohistochemistry, and then we also collect additional slices for proteomics and electron microscopy. And I'm hoping for the future years to present you data on those. But this is an example image of where these biopsies are coming from. They're really small. They are 3 × 3 millimeter in size. So there's not much we can do with it, but we still, if you cut them and do a Nissl stain, as seen in this picture, you can appreciate that we preserve all the layers of the cortex, starting from the pia up until the white matter, so that we can now start looking at things in a layer-specific manner, so that we can start asking or answering some of the fundamental questions in the field of Alzheimer's disease, such as how does early AD-related pathology, beta amyloid or tau, alter either single neuron or neural network operational properties? And how does this pathology then alter neuronal morphological parameters? So we have been doing this for quite many years, and as presented by Evan, with that sample set that was published in that Cell paper, we had, at that time, we were really excited about the data. We had a number of about 8 or 9 in our database at that time. But since then, throughout the years, we have collected more samples, and now our database is much more extensive. And what I'm really happy to say is that we're able to reproduce the same data as we published with Evan's paper, showing that the biopsies, especially with the low A-beta load, showed a hyperexcitable phenotype that was evident in an increased number of spiking and bursting parameters in response to NMDA activation. However, what is really interesting is that, well, back then we did not really have more than a few cases with tau, a co-additional tau pathology. But right now in our database, we have a significant number of patients that have both A-beta and tau pathology, so that we're able to look at how the concurrent tau in these biopsies alter these neural functional properties. So we have reanalyzed our data set and divided now the samples to samples that do not have any pathology, and then the ones that have beta amyloid pathology only, shown here in this graph E in blue, and then the ones that have both amyloid and tau pathology. And that graph shows the extent of beta amyloid load in these biopsies, whereby you can kind of appreciate that there is quite a lot of variability in the amyloid load in these biopsies. But what is really interesting is that by analyzing the similar parameters as what we did before, we were able to show that co-occurrence of tau in these biopsies also contributes to the hyperexcitable NMDA-induced phenotype that is evident in the percentage of bursting electrodes, as well as some additional bursting parameters. But however, this hyperexcitability is quite different between the beta amyloid and tau patients, in a way that the coefficient of variation of the interspike interval is actually decreased in the beta amyloid patients, whereas with the tau carriers, it is increased. And this might suggest that individuals with tau show deficits either in the spike timing or then the recovery dynamics of the network. So we also did patch clamp studies in order to really understand the intraneuronal properties of these neurons. And our data kind of support the metadata that we have in a way that we see now specifically in the tau-carrying patients, alterations in these neural properties that are not evident in the patients that have beta amyloid only. In a way that we see a significant decrease in the capacitance of the tau-carrying patients, shown here in I don't know where my cursor is, unfortunately, but shown here in the graph B, as well as increases in the sodium and potassium currents in these tau-carrying neurons, as well as increase in the action potential amplitudes. And now, really going hand in hand with increased potassium flux in these neurons, we also see increase in the after hyperpolarization. And if you look at the traces and we calculate the area under the curve of the after hyperpolarization, we see an increase in there as well, kind of meaning that the after firing and action potential, it takes more time for these tau-positive neurons to recover from this action potential firing. And since Evans' data showed very interesting findings on the lack of inhibitory interneurons in the layer 1, we set up to kind of investigate whether we can have a functional correlation with his data. So we did an experiment in which we patched a neuron in the layer 2, 3, and we placed a stimulating electrode in the layer 1. And we injected current into the neuron to make it fire action potential. Showing these representatives graph on the right, you can see that you have action potential firing, and whenever we stimulate the layer 1, you have a cessation in this firing, suggesting that there is an inhibitory input from the layer 1 to the deep layer of the neuron, of the cortex. However, in the beta amyloid positive cases, this kind of inhibition is lacking. And below is a quantification of this data, and this is not evident anymore with the patient that have tau pathology. And every time when we patch a neuron, we also fill it with biocidin so that we can actually look at neuronal morphological characteristics. And after this quantification, we can also stay in the sections with beta amyloid, shown here in the right, so that we can actually take a look how beta amyloid deposits alter neuronal morphological characteristics. And we have done an extensive quantification of different parameters of the neurons. Here are some of the patched neurons shown. And very interestingly, where we see significant differences is that the number of spines in these neurons is actually increased. And this is true for both the beta amyloid as well as beta amyloid and tau positive cases. And it's very specific for the apical dendrites. Now, we were also, since we had this very striking or unexpected increase in the number of spines in these biopsies, we also took a look on what happens to the spine densities when this dendrite crosses an A-beta deposit. So we were calculating spines before the plaque, then at the contact with the plaque and after, and saw that there is this is not a significant trend because there's not many dendrites that actually cross a plaque. This is a very random process in these. But we were able to see a slight decrease with this. And the spine stains with both excitatory and inhibitory markers suggesting that they might be functional. So we wanted to find some correlations on what happens in the brain that could explain what we see in our electrophysiology as well as the morphological reconstructions. So we did an untargeted proteomics of these brain samples. This is total brain proteomics. And what we saw that what was kind of nicely in line with our data was that a lot of the proteins that are related to synaptic transmission, synaptic physical trafficking, and, for example, glutamatergic signaling are actually increased in both the beta amyloid and tau positive cases. Then we went into reanalyzing the single-set data set that was generated by Evans' lab. We took a look at specifically the excitatory neurons in the layer 2, in these biopsies, and we were able to identify 14 different excitatory neuron clusters from which specific cluster number 9 showed the greatest enrichment of different pathways. And these pathways were related to synaptogenesis, synaptic signaling, and AMPA and glutamate receptor pathways. And we were able to define that these neurons were RORP positive. And that last graph showed quite many deregulated genes that are related with the synaptic signaling in these neurons. So then we did spatial transcriptomics using the Xenium technology. And we were able to identify several cellular populations in these biopsies. And the graphs G shows the cluster map of where these neurons reside. And the RORP positive cells that we believe that we have been patching are indeed located in the layer 2 to 3. And after the Xenium, we have stained the samples with beta amyloid. And now together with Beth and Evan, we're aiming to see how these altered RORP positive cells interact with microglial states and whether this is altered based on pathology. And since our electrophysiological signatures were quite striking, we wanted to ask, could we predict the patient pathology based on the electrophysiological signatures and the associated metadata? And we used random forest-based classification tool. And indeed, we were able to very nicely classify these patients into different pathology groups based on these properties. So basically, the take-home message, what we have is that the early AD-related pathology causes alterations, hyperexcitable phenotype in these biopsies, but it's very different between the patients that have beta amyloid or tau pathology. Whereas beta amyloid alone seems to drive glutamatergic hyperexcitability, the addition of tau in these biopsies seems to cause single neuron operational alterations in the operational deficits. And we have this surprising increase in spines. So yeah, with that, I want to thank you, all our collaborators in my lab, and also for your attention.
Thank you, Tarja. Our next speaker is Sam Marsh, presenting on behalf of Beth Stevens' team. He's going to compare paired samples of CSF and blood to map immune cell states from each of these to their functions. Sam.
Thank you, and thank all of you for coming today. I'm really excited to share some of this new data that we've been generating from this unique cohort and really looking at immune populations in the CSF, which represents a really accessible compartment, not just in this cohort, but in other cohorts as well. What are we using to advance? That one, there we go. Nothing to disclose. And a quick thank you. You've already heard excellent talks from our other collaborators here, and you're going to be hearing from Henrik after me, but this really is a true group effort and really wouldn't be possible without everybody involved. So we've heard some really excellent talks from Ville and from Evan and from Tarja about different characteristics that we can look at with the brain biopsies from these patients. But one of the other things that we're really interested in is moving beyond the brain parenchyma and what we can learn from other areas of the CNS. And one of those areas that we're really interested in are these areas of neuroimmune interaction. And so we can look at things like the brain lymphatic system or perivascular and border spaces. But what I'm really interested in is the CSF. And the CSF is home to a diverse array of resident immune populations, but they are very rare. We're talking about 1 to 5 cells per microliter. And so studying them has been very difficult, but single-cell sequencing has really opened the door for our ability to learn about these cells and learn what might be going on in the CSF. And so with that, now that we can profile these cells, we're also obviously interested in how these cells might change over the course of disease and what that means for the progression and the development of the disease. And as we've talked about, INPH provides this really unique early window because not only do we have these CSF cells, but we also know what's going on with the brain transcriptomes. And then that allows us to do correlations between those to really understand what interactions might be taking place. So this is just a quick demographic overview of the INPH patients that we have here. But one of the other questions that we went into this, especially with the CSF, is how does INPH differ from healthy controls if they don't have any pathology? And the same, how do INPH patients that have amyloid or tau pathology compare to true AD patients? And so to get a better handle on this, we have a collaboration with Henrik Zetterberg and Silke Kern from University of Gothenburg, and we also have 32 patients from their memory clinic who are control or AD patients that we can also include in this study. And I don't have time to go into the results of a lot of the comparisons that we've done, but it's looking very similar between the cohorts. So we really think that we can use INPH in a lot of ways to look at these questions. And so a lot of the data that I'm going to be sharing with you is the combination of them both. And one other thing just to plug here, because it's something that we started after we started the collaboration with Ville, is that we've spent a lot of time optimizing the protocols for cryopreservation and for processing of these cells after cryopreservation before sequencing, and critically in a serum-free manner, because serum and immune cells don't mix very well, especially in cells never exposed to serum in real life. And so this protocol is really easy to adapt and to implement in multiple centers, and we're really happy to share this wide. And we've been getting much better yield of immune cells from our protocol, as well as much better quality of the transcriptomes when we do the sequencing. So this is the overall landscape of our current data set. We have 98 samples, about 156,000 cells. We see all of the kind of expected populations, and they display kind of the canonical gene expression patterns that we would expect from T cells or from B cells or from monocytes. But a lot of the focus so far in CSF studies with single-cell sequencing has been on adaptive immune populations, on the T cells especially. And while we are interested in the T cells and we have ongoing studies there, we were really interested in the myeloid populations, and especially because due to the increased yield in our protocol, we have many more times myeloid cells than previous published single-cell CSF studies. And so we're really powered to look into these populations. And so in the blue color there on the right, you can see the kind of their classical dendritic cells. We see our CD14 monocytes, and you see I have a population that initially when we started this study, I labeled as CSF myeloid. Now, these are cells that were typically labeled in previous studies as CD16 or non-canonical monocytes. Some people called them microglia-like cells. I can assure you that it's neither of those two things. But we really still didn't have a handle on what they are. And I think part of what I'm going to be telling you about is why we think that these are actually a very unique population of macrophages that are specific to the CSF. So this is just a subclustering now of all of the cells that we see there. We have our macrophages, our dendritic cells, our monocytes. We also see a small population of intermediate cells that we believe are a transitory population of monocyte differentiation into final lineages, which corresponds to kind of adult myelopoiesis that you see throughout the body. One of the ways that we can look at and try and identify, what these cells might be is by doing gene module scoring. So instead of looking at the expression of one single gene, we can look at whether a set of genes is enriched versus a set of controlled genes in every particular cell. And so just as an example here, we took a set of genes that define CD14 monocytes, and we looked for enrichment, and not to very surprise, we see this enrichment in the monocyte cluster there on the left. But this kind of, validates that we're looking at the right thing. When we do the same thing with a set of genes that define CD16 monocytes, we see little to no expression basically anywhere within our clusters. And so this really helped us, further, convince us that these weren't, CD16 monocytes. And when we use these same scores in the paired blood from the patients, we see nice enrichment in both the CD16 and CD14 populations that are both present in the blood. So what are these cells? Well, they did express a number of genes that we kind of associate with typical macrophages. And so we took a score of that represents a kind of conventional tissue macrophage score, and we used that on the cells. And you can see it does enrich in this cluster of CSF macrophages, but it wasn't the kind of dramatic, whole enrichment that we were really expecting. But one of the unique things about these cells compared to other macrophages is that they're free-floating. So these are cells floating in the ventricles, in the CSF that's present throughout the body, and most other macrophages are in extracellular spaces. They're closely associated with other cells, and so they're not in this atmosphere. But there is actually another population of cells in the body that are free-floating, that are macrophages. And these are the peritoneal cavity macrophages. This is a paper from Gwen Randolph's lab. And so we wondered whether our cells might be another type of cavity macrophages, and the CSF is simply another cavity. And so what we did is we took the genes that Gwen's group identified in their paper that defined these cavity macrophages, and we did this same scoring. And what we find is now this dramatic enrichment of that gene set in our macrophages. And so this really leads us to believe that what we're looking at here is a unique population of cavity macrophages specific to the CSF. And again, I don't have the time we have here to go into it, but we've also been digging deeper into the heterogeneity of these clusters, and I can say that we have a number of different subpopulations within the CSF macrophage cluster, and we're doing more work to kind of elucidate the differences between these and what the functional impact of these different populations might be. So now that we've identified these cells, the question becomes, well, are they relevant to neurodegenerative disease? And I think you know the answer, because I wouldn't be here telling you about them if they weren't relevant to neurodegenerative disease. And so this is a plot that, I'm sure you've seen many times in the conference. These are the GWAS loci identified in AD. And one of the things that we can do with single-cell sequencing is we can look for enrichment of the heritability of those loci in each individual cell. And so this is an algorithm called SCDRS, which was published a couple years ago in Nature Genetics, which allows you to do this scoring from any summary statistics from a GWAS. And this is the result. We see this huge, very dramatic enrichment of AD heritability in these CSF macrophages. We also see it to some extent in the monocytes and the DCs, but it is dramatically more so in these macrophages. And so this really, puts to the point that these cells might be relevant to some aspect of the disease because they're expressing a lot of these genes. And so the question then becomes, well, how do these cells change during the course of neurodegeneration? Well, I'm sure you've also seen many plots on the A-beta 42/40 ratio throughout this conference. And so we do see, this is in the INPH patients, the associated drop in the INPH ratio with the positivity of either A-beta tau, or tau-only pathology identified in the brain biopsy slice. So that's what's on the x-axis there. Now, even in a cohort this size, we have over 60 INPH patients included in this. These kind of discrete categories still present a problem for differential analyses like differential abundance and differential expression, because they may not capture the heterogeneity within those populations. You can see, especially in the A-beta and the A-beta tau groups, there is considerable variability in the ratios that we see for A-beta here. But what we can do is we can model the A-beta 42/40 ratio as a continuous variable and look for changes that are in abundance that are associated with this ratio. So this is work that has been led and collaborated with Vishal Sarsani and Evan's group, where we can see a number of these CSF macrophage populations that show a decrease in their abundance with the decreased A-beta 42/40 ratio. Interestingly, we also see some increases in the CDC23s and the CD14 monocytes with changes in this ratio. We can do the same type of analysis, but instead now looking at differentially expressed genes in the subclusters. So these are differentially either up or down regulated genes that we now find are also associated with the ratio changes in these patients. And so right now we're in the process of digging deeper into the gene sets that we've identified in each of these clusters to really get a better sense of what pathways and functions might be active in these cells and how this change might be affecting things. We also, again, for the sake of time, I don't have time to really go into it. We're writing a preprint, which hopefully we'll have out in the fall, but we've done a lot of deeper analysis too, looking at transcription factor networks, gene regulatory networks, co-expression modules, and we're finding a number of interesting things that really define these cells and make them unique from the other myeloid populations in the CSF. And so just to kind of close on kind of what we're looking at in terms of future directions with these cells, now that we've identified these cells, it's really important to consider them as macrophages, because if they're being labeled as monocytes, your interpretation of a change in a certain pathway is going to be very different based on what we know the functions of a monocyte versus a macrophage. So we're developing right now a spectral flow cytometry panel that will allow us to validate a lot of these results at the protein level. Will allow us also to sort these cells out from CSF samples and allow us for a faster, easier, and much cheaper way to analyze these cells. We are also really interested in performing an assay that our lab developed called FEAST, which is a flow cytometry high throughput assay to understand what myeloid cells are eating. And this is a very detailed protocol. It came out in Nature Communications 2 years ago. And so we're really interested to look at, are these cells engulfing amyloid? Are they engulfing tau? Are they engulfing neuronal or synaptic proteins like neurofilament light that are present in the CSF? And potentially down the line, are these different between the groups? And so, and again, given the enrichment of the AD heritability here, we really want to know, can we learn more about their function and how is their function changing over time? And finally, just to kind of close on some bigger picture here, we're really excited about the ability now to bring the CSF and the proteomics and all of this rich clinical data together, because it's really, that integrative analysis is really going to allow us to understand some fundamental questions that are not possible to do in other cohorts. How do we identify markers of onset and progression of AD? What do the fluid biomarkers tell us about cellular functions that are changing in the disease? And importantly for these CSF macrophages, how might they be interacting with other immune cells in the brain, in the blood, or we've also started some analyses on dural meninges, and so we're really excited to dig into that. I want to thank everybody involved in this study. A big thank you especially to my mentor, Beth Stevens. A big thank you also to Anna Kane, who's an associate director in our group, who really makes all of our work in the lab possible. Evan's lab, Clary and Neer as well at the Broad, Henrik and Silke in Sweden for facilitating the memory clinic studies, Ville, who none of this would be possible without Ville. So Ville is our unsung hero of everything. And I just, especially our funding, as Alzheimer's Association has been wonderful in funding this work. And I want to add as a final thing, I'm on the lookout for new job opportunities in industry and biotech. So if you're interested, please reach out. My email's in the slide, or come find me after the talk. I'd love to chat more. Thank you.
Thank you very much, Sam. And also, I think Beth is there? Oh, yeah, she's at the end, right? Yeah, so
Yeah.
Beth is here also to answer any questions that you have related to this work. And now, continuing on the CSF journey, it's my pleasure to introduce our last speaker, Dr. Henrik Zetterberg, and he's going to describe CSF proteomics changes in IMPH patients with and without AD neuropathology.
Thank you very much. Thanks for being here, and thanks for the invitation to present. When I listened to all these talks, one thing struck me, because what you presented, Sam, comes from a source of biological material that we used to throw away in the lab. And it's a bit, when you think about it, it doesn't make sense at all. So when you do a diagnostic CSF sampling, you take out 10 milliliters of CSF. It's a simple procedure. It's also not that difficult for the patients most of the time. So it's really something one could consider doing. And now in the era of blood biomarkers, one could ask if the CSF-based research really will continue. But I think it's a good, as you have heard, a really good source for biomarker discovery. So I think we should continue in research to do this. When we get the sample to the lab, we spin it, 2000 G, and then we take the supernatant for biomarker analysis and we throw away the cell pellet, which feels like such a shame. Instead of doing that, you can simply, when you have taken the supernatant, add some cryostore and put this in -80, and then you can keep the sample for SAM. So, and it's super easy to do it. And so I would encourage all of you who do CSF-based research to save the cell pellet, and we can share the protocol also for how to do it. And then Sam will analyze the samples for you. And I mean, the same goes for Ville's practice here also, with actually making use of the surgical procedure to also allow patients to contribute material for important research. Here are my disclosures. So what have we learned about amyloid biomarkers thanks to this work? Ville and I, we started to collaborate in 2009 or something like that. And then this is an old study that we did together on consecutive IMPH patients who underwent evaluation for shunt therapy and eventually had both shunt placement, pre-shunt lumbar puncture and post-shunt lumbar puncture. And then we could start to look at different relationships between A-beta levels in biofluids and compare lumbar and ventricular levels, and also, of course, the brain biopsy diagnosis. So we got to know that lumbar and ventricular CSF levels of A-beta 42, they correlate. We could also see that the more amyloid pathology in the biopsy, the lower CSF A-beta 42 concentration is in both ventricular and lumbar CSF. A little bit noisy, but still important information. And then this turned out to be an important finding. We knew for the whole IMPH field, knew that CSF beta 42 concentrations tended to be low, and for a while there was a view that INPH increased the risk of Alzheimer's disease because of induction of amyloid pathology. But what we could see that through these studies is that in INPH you have a general lowering of A beta peptide levels, 38, 40, and 42, but 42 is the only one indicating amyloid pathology. And that made people start to think of using ratios. So A beta 42 over 40 became a more close amyloid plaque pathology marker than A beta 42 alone. And this general reduction of CSF A beta 38, 40, and 42 seen in INPH was what explained the thought that INPH could induce amyloid pathophysiology. The ratio showed that the prevalence of 70-year-old people with INPH, the prevalence of amyloid pathology was 20%, which is similar to the general population. And to some extent, that could be important for the studies we are doing here. Most likely, this is not a special form of Alzheimer's that is studied in Wille samples. It is Alzheimer's disease, but not induced by a CSF dynamics disturbance disorder. And the associations with ApoE and so seems to be similar. And this is now the FDA-approved CSF-based test for amyloid plaque pathology, the A beta 42 over 40 amyloid ratio, almost 100% concordant with amyloid PET. In clinical practice, it looks like this. You get two normal distributions of results, which is very unusual for a clinical chemistry test. And if one looks in the right part of this graph, this is general clinical chemistry results, 12,000 results, completely unsorted, and you always get this type of bimodal distribution. The cut point is the same in INPH as in memory clinic patients without INPH. Then we have also started to do biomarker discovery in these pressure samples, and this is work by Johan Gobom and Sofia Weiner in my team in Gothenburg. And I will show you some snapshots. These are TMT proteomic data sets. So it's tandem mass tag proteomics, where we first take the CSF sample, separate peptides from proteins. We label the peptides with tandem mass tags that can be looked at isobaric tags of peptides that we could then differentiate with mass spectrometry. And then we also take the proteins and digest with trypsin and label with TMT regions. This is the setup of the workflow. And here are just a couple of results that I want to highlight. When we have done this in Wille's samples and separated the patients according to whether or not the brain biopsy has amyloid, tau, or both, or neither, I should say, then we get a synaptic biomarker signal. So we see presynaptic markers being upregulated, especially in A+, T+ individuals. And we also see, which is a replicated finding, it was known from before, a reduction in NPTX2. And this ratio seems to be a relatively important ratio in terms of determining who are the amyloid-positive people likely to progress. And this ratio seems to help to tell that. So we have seen it in several other proteomics data sets. This is a targeted panel developed by Johanna Nilsson, who just defended her thesis in Gothenburg, where we see again 1433, which is the presynaptic marker, and NPTX2, which is the transsynaptic marker that is reduced across neurodegenerative diseases. So 1433 up, NPTX2 down. And if we look at the ratio here, it turns out that this seems to be some type of synaptic resilience index. So here you see people who are in different levels of abnormality for this ratio, and those who have a normal ratio don't progress over more than 5 years. And these are data from the Biofinder study. With Tony Weiss-Correa and his PhD student Hamilton O, who now also has defended his thesis and as a postdoc in New York, I think, they also had similar data. And this is what it looks like. If one takes the classical thing, comparing amyloid and tau positive individuals with amyloid and tau negative individuals, and look at clinical progression, in this case over 10 to 15 years, you see that, of course, the amyloid and tau positive people are the ones progressing the fastest. But if we instead look at this ratio, we see almost a clearer separation between fast versus slow progressors. And if you have both amyloid tau and a synaptic index, which is abnormal, it goes super fast. So this could potentially help us if we want to characterize people according to who are likely to, or who are in great need to start on amyloid targeting therapy. This index could be an interesting index to look at. It might be that plasma phospho-tau tells a similar story and that it's not needed, but it could still be interesting, I think. So to conclude this work, brain biopsy data on A-beta and tau as the reference standard has basically helped to discover a novel synaptic biomarker index. And this normal synaptic index can tell if people are likely to progress or not if they have amyloid. A similar approach will be used for microglia and astrocytes, of course, to see if we could find beneficial or detrimental activation patterns of these molecules. And the brain biopsy data will make it possible for us to examine the CSF proteome in relation to reference standards that are not accessible by other means. Simply, perhaps I shouldn't say by staining for different types of molecular phenotypes, cellular phenotypes in the brain biopsy. Brain biopsy staining can provide novel reference standards for fluid biomarker discovery. And the limitation, of course, which we also can discuss, is that we are in the frontal lobe and we look at a very small part of the brain. But I think people in the panel have lots of good reasons to defend the approach. Thank you very much.
Thank you very much, Henrik. So now we are going to open the floor for questions and discussions. So we have one question online that is going to be for Sam and Beth. Can you tell us more about findings of the macrophage population that you have described?
Generally, yes, we can tell you lots about it. If you want to reach out, whoever it is, directly, we're happy to answer any questions. I think it's really interesting to us because it represents this population that has been understudied. And I think, the optimization of the protocols that allow us to get better recovery of these cells really facilitate now a better ability to understand it. And as Henrik mentioned, the ability to implement these protocols to save what is normally a discarded CSF pellet, hopefully that can be implemented more and more, and having numbers in this type of study when we can approach the numbers that we have for the brains that Evan's doing are, I think, really going to help us better understand these cells even more.
Beth, do you want to add anything to that? You're good? Okay, we're going to start with our first question at microphone 2, please. Please identify yourself.
Hi, I'm Walter Korshutz from NIH. I had a question about something I should know, but I don't. The CSF macrophages that you described, are they necessarily a different type or potentially a different state? And the question is, could those macrophages have entered the brain, then moved into the CSF? And do you see any signs of that, or do you think they're a completely different type that came in from the meninges?
Yeah, I think that's a really good question. I think that's something that we'd like to look at more. Our assumption, and I think the assumption based on what we know and what the literature knows from the peritoneal cavity, is that these are a resident population in the CSF that come from monocyte differentiation throughout life. And I think it's definitely possible that they could be recruited to the brain. That's one of the potential hypotheses for why we're seeing a reduction in these populations when we see more amyloid in the brain. It's possible that maybe these cells are actually being recruited out of the CSF to the choroid plexus, to the brain. So I think that's something to look at. Unfortunately, in a lot of the preclinical models, it's very difficult to study these kind of cells because we're talking about 1 to 5 cells per microliter, and so the amount of CSF you can get from a mouse and a rat is pretty limiting, and so we're limited in a lot of cases to the human samples. But I think it's something that we definitely want to look at. Now that we have better signatures, we can look at this a little bit more when we look at cells from the brain.
Microphone 3, please.
Thank you. Fiona Elwood, J&J. Great set of talks, really fantastic. My question is for Evan. The 2 different microglial cell types that you looked at, you showed the ratio and how that's separated, high ratio, low ratio, and how that separated with the progression. But what would happen if you just looked at the dam state on its own, high versus low, or the homeostatic state, high versus low? Did that also separate? Did those curves also separate as much?
Yeah, it also separates, but the ratio is much better powered. So I think, and I think it kind of makes sense because they're both expanding. So you're identifying some reactive state, but the hypothesis we have is that there's a sort of predisposition to push in one direction or the other. So by looking at the ratio, just better powered. And we've also seen that the ratio is better powered for genetics as well versus just the proportion of the individual populations.
Microphone number 5.
Hi, Isabel Castaigne from BIDMC and Harvard Medical School. First of all, I am so excited about all the work you're doing. I think this is going to be such a huge resource for the field. Thank you so much for doing it. My question is for Sam, also huge fan of your work. So you did touch on it. I already had the question, and then you had a slide sort of showing the question. And I do understand that it's a lot of speculation, but if you could predict, what are your hypotheses of what are these macrophages doing, and how do you connect what you're seeing in the CSF to what is happening in the brain?
That's a great question. I think that's the million-dollar question with these cells for sure. I think that there's a lot of things that we can hypothesize. I think the being able to do the feast assay, I think, is going to be really informative for us overall to understand whether there's kind of a steady state engulfment of substrates in the CSF by these cells. Taking what we know from the peritoneal cavity and taking what is also kind of known about other the T cells and things in the CSF, I think they're likely performing a general surveillance role. For instance, in the peritoneal cavity, in the cases of inflammation, they respond very heavily to viral infection and things like that. In the case of bleeds or kind of leakage into the cavity, they're extremely good at scavenging red blood cells and hemoglobin. And so I think that's potentially another function that they're there as kind of a stopgap surveillance. And I think that's something that we really hope to learn, and I think we hope to be able to show the importance of these cells so that more and more people study them and we find out more about them.
Can I just add one comment to that? I think Sam's point about what they're eating and what they're scavenging is one question we are really interested in, not just because of this result, but because as we start to study this even in animal models, we're appreciating that the border-associated macrophages and macrophages associated with choroid plexus, they're different than the microglia. They have these different scavenging type receptors. So now that we know that, now we have ways to study that and model that. We can then ask, how does genetics change that? You saw them enriched in all of these AD risk genes. What happens in different genotypes? These are the kinds of functional assays with FEAST and some of these other things that we can do with microglia or even iPS cells. We're hoping to be able to enrich for these cells and study them in a more functional way, as Sam mentioned. But the scavenging also goes down with aging, normally, at least with brain border cells. So that might be another source of either vulnerability or resilience if these cells aren't scavenging the way they should, right, in the context of an aged environment.
Thank you.
Microphone 6.
Hi, Adam Abdulrahman from Vanderbilt University. I have another question for you, Sam. Do these CSF macrophages seem to have, is their transcriptome close to any other border-associated macrophage populations? Are they close to epiplexus, choroid epiplexus BAMs?
So at least from what we can tell, they do look, there are shared similarities. It's why we kind of thought that they could potentially be a macrophage subtype in general. They're very high for TREM2 and ApoE. They're high for C1q, kind of all the components there. But they are much lower than you would expect for things like MRC1, CD206. LIV1 is not quite as high either. When we do that same gene module scoring that I showed using the conventional macrophage score on perivascular macrophages that come from Evan's single nucleus brain data set, those macrophages show a dramatic enrichment for that conventional score. And so they do look, like we would expect a conventional tissue macrophage to look. So there are a lot of similarities, but there are also some key differences.
Thank you. Microphone 3. Microscope 3, please.
I'll be quick, or we can take it afterwards. Really impressive work, and I'm really excited about the data sets. I think this is really going to be training. It's a dream cohort. Again, I'm Tawfiq Raj from Mount Sinai, New York. My question, I guess, maybe you guys are doing it already, looking at genetics. And Beth already mentioned Sam and Evan. Maybe you can comment on QTLs, right? We've seen QTLs that show up in both monocytes and peripheral monocytes and microglia. I wonder if this kind of data sets can help us, because as you all know, most of the AD risk variants have effect in both monocytes and microglia, and it's been difficult to tease that apart. So do you think your data set, I mean, are you guys thinking about doing QTLs? Because I think you have power, right?
Yeah, I don't know. Evans can speak to the brain. I don't know, how powered we're going to be in the CSF, just given our sample number. But we do have, I think it's 600,000 or 700,000 cells from paired blood from these patients. So we are, that's going to be better powered for that kind of analysis. And we do have whole genome on all of them. So I think that's something that we're very interested in. And I think that's something we'll definitely take a look at.
Yeah, we're really loving these eQTL and proportional QTL analyses, because I think there's a few things that are really instructive about them. The first thing is that it kind of validates that there's something biological happening whatever you're testing, because the sort of technical or null hypothesis of a QTL analysis is just going to give you no signal, right? So if you get more signal, then it could be it's likely to be a biological. So I mentioned the proportion analysis as being very useful for that, to sort of identify that the ratio of these two populations seems to be connoting something more biological than just the individual population by itself. But the other thing that we've been looking at is also just the strength of QTLs, in particular AD-associated genes across different microglial subtypes. And I think that is where we can also really start to inform processes by which these particular genes are actually acting in particular subsets of cells. And we do see differences, and it's not just driven by power. So the largest cell types, like the homeostatic microglia, which occupy like 80% of the cells, they don't always have the strongest QTLs for PCALM or all these other microglial genes. And so we think this is a window into identifying how to sort of parse out individual QTL subsets into functional groups.
And you are enriching for it, right, for microglia in the brain?
We're not enriching for it. The other interesting thing about I could talk a lot about this. The other interesting thing about this is in the NPH cohort, the microglia, and actually the glia in general, have stronger eQTL signals than the postmortem tissue. And I think that makes a lot of sense, because as actually Sam has shown in his first work, the glial cells are extremely prone to technical artifacts associated with death, right? Activation and different kinds of signatures. And I think that just basically dilutes your QTL power relative to
You've seen the same in
You've seen the same thing.
yeah.
Yeah, very cool.
Great, thank you.
Microphone 6, please.
Hi, thank you. Thanks for a great session. Jo Jackson from Imperial College. I've got a question for Henrik about your synaptic biomarkers. So do you know what is driving that ratio? Is it a change in synapse number or a change in the health of the remaining synapses?
Yep, we don't know yet, but we have done some studies in a cohort where there was SV2A PET imaging, and then there was the lower SV2 PET signal, the lower NPTX2, for example. The presynaptic markers, I think actually there is some type of hyperexcitability contributing to increased release, and it also correlates with phospho-tau increase, but we don't know exactly what the mechanisms are.
Sure, thank you.
We have a couple of questions online for Sam and Beth. In blood, did you find similar patterns of your myeloid populations? And also in your CSF data, did you identify neutrophil populations?
So the second one is easy. We don't see any neutrophils. Neutrophils just in general for sequencing are very hard to capture. They just kind of burst and die right away. It's why you often see neutrophil genes as just ambient RNA contamination in all of these studies. You'll see these, really highly. For the second question, whether we see this in blood, this population is not present in blood. So blood just contains kind of our normal monocyte populations. We do see the CD16 populations. I will say they're not in this analysis because they were removed, but we have samples when we processed them, we noted a blood coloring in the CSF in the pellet that we got, and so those are removed from the analysis. But we still sequenced them to see if we could see differences or what might be going on. And in those ones, we can see true CD16 monocytes because they're likely just coming in from the blood and they're contaminating that sample. They're very, very small. One of the things that we're looking at right now, and it finished like 5 days before we came here, is we've just done some consensus NMF analysis, both on the blood and now on the CSF. And so we're really interested in looking at whether we can see modules and factors from that NMF that show either overlap between the blood and the CSF, or again, ones that might correlate with some of the different biomarker changes that we're looking at.
Thank you. Microphone 5, please.
Thank you. Betty Times from Amsterdam. I have a question for Tarja. I think your electrophysiology measures really give this additional layer in giving us information that I at least really wanted to know, because you're showing that now you have more excitability when there's amyloid and tau. And so I was wondering, are you also measuring maybe amyloid and tau and perhaps other protein measures dynamically, or do you have plans? Is this possible? Can we now finally test this hypothesis in human tissue?
So you were asking like if we use other stimuli except for the NMDA?
Yeah.
Yes. So we do. So we have so far routinely exposed so we have perfusion system equipped with our MEA. So we are doing pharmacology for NMDA, but also for carbachol, so that we can also look at the physics in the cholinergic system. So these are data that we have not yet touched and analyzed. So we have a lot to do in that regard.
Okay, so we have to be patient.
It would also be super interesting, of course, to co-correlate the measurements on the synaptic markers and phospholipid with your markers, and we haven't done that yet.
Exactly, yeah.
Microphone 4.
Yeah, thank you very much. Marcel Wu from Hamburg, Germany. I have a question to Dr. Zetterberg, since you mentioned this easy centrifugation protocol. I was wondering whether you have any idea whether these proteins are really in the supernatant or whether they are actually transported by extracellular vesicles or some kind of other organelles.
Yep, those of you who have been working with EVs will know that CSF is very relatively low in EVs compared with blood. That could be a good thing because 80% of the EVs in blood might be platelet derived and perhaps not super interesting to what we are trying to examine. The protocol we have used for SAM's cells is we didn't change anything in our clinical practice. We simply spin the sample 2000 G for 10 minutes at room temperature, and that will not make any EVs end up in the pellet. So if there are EVs, they will likely be in the supernatant. So the cells will contain cells, and the fluid that we do the proteomics on will actually contain some EVs, but it's not much in lumbar CSF, actually. So you would probably have to do a protocol to enrich. If you're specifically interested in EVs, I think one needs to spin again, actually.
Tarja, did you want to mention the plans for EVs?
Yeah, so we are we're also interested in isolating EVs or enriching EVs in these samples and doing either transcriptomics or proteomics, but this is work ongoing, so.
Stay tuned. Number 6, please.
Hi, this is Lawrence Shea from J&J. This question is for Tarja and Evans. I'm highly interested in the reason why you both observed the layer 1 interneuron loss, and I was wondering if you can comment on whether this is a cell-autonomous effect from the interneurons themselves, or could it be a non-cell-autonomous from the microglia, especially the LDL CD83 positive cohort?
The answer is we don't know. We're still trying to understand the exact relationship between the selective depletion and the different cohorts that we've examined it in. The same population, we saw the depletion of the same population in the ROSMAP cohort, but actually in larger sample sizes of the ROSMAP cohort, it was more closely associated with cognitive loss than it was associated with amyloid. So we're not quite sure exactly what the relationship is between, what the explanation is here. It's clearly like an important population, but we don't quite understand it. And I think our two major hypotheses about why this population might be selectively depleted is, first, there's clearly some sort of superficial cortical vulnerability to AD that's been observed. I think read papers, Brad Hyman papers from 1980s showing selective depletion of upper layer, layer 2-3 excitatory cells. So I think there is a clear bias towards superficial loss that's been observed many times. So it could be something to do with the circuitry, right? Maybe inputs from from cholinergic cells, or the second possibility I think is it has something to do with actual penetration. I was just at a session on neuropathology in the last hour, and we saw some initial presentation on ARIA samples and depleted, aducanumab depleted pathology samples, and there's a clear depletion in layer 1, which was like very surprising to me. That was more preferentially cleared than the other layers. So there's something interesting about layer 1, but we don't quite understand what it is.
Dr. Koroshetz.
Sorry, I'm okay. Fixate on this macrophage stuff. So I guess the question is, do you know if you looked at markers that would differentiate bone marrow derived from yolk sac derived? Because it sounds like from ChatGPT, in the peritoneum you get both. Yes. I guess if it's yolk sac derived, then same with the microglia. So maybe the genes that you see heavily expressed are not that surprising if they're coming from a microglial lineage.
Yeah, I think from the peritoneal cavity, the initial seeding of the cavity is from the yolk sac myelopoiesis, and I think some from fetal liver. But then over time, that basically gets turned over, and especially there's evidence that basically if there's any insult to the peritoneal cavity, those initial ones get depleted very quickly, and then the monocyte-derived ones take over. I think there's a really good opportunity to kind of look at this more. We do see a very small population of cells in the CSF that look a bit more like the conventional macrophage cells, and those either could be longer-lived cells coming off of the ventricle wall or coming off of the borders, because I think it's very likely, I think, that the initial seeding of the compartment matches what we know about the border macrophages and about microglia, that it comes from the yolk sac. But then I think, again, once we're talking about, especially in this population where we're 65 plus and we're talking about, patients that have likely had many insults over time, it's likely that I think they've turned over from the monocytes.
Just a comment, and we'd love to know the origin. But the other thing is, if we can also obtain choroid plexus samples, we can also go back and try to think about where they reside. Are they in the ventricles? Are in the choroid plexus? So these are some of the things we're thinking about. Because with the markers now that Sam's identified, that would enable us, it won't get you at origin necessarily, but it would give you a spatial look at where these cells may reside in human tissue outside of IMPH. And also, maybe we could, if we knew how, pick these up in animal models where we could do fate mapping. So that's really we've been talking to Maria Lettinen and others that study the choroid plexus as a way of doing that.
Yeah. And I think there's potentially an opportunity in some of the larger animal models. I know Maria's lab is starting to work with some porcine models, or some of the non-human primate models. If we're, once we're more confident in what we're looking at, and we could really have a target to go after, I think that would be something interesting to look at in the future.
All right, it seems that there are no more questions online or here, so I just want to open it for the panel in case there is any questions that you would like to do to the rest of the speakers.
I have a question for anyone who wants to answer this. Clearly, amyloid and tau walk hand in hand in some of these biopsies. Do you think any of these technologies might be useful for maybe finding out more about other co-pathologies like TDP43, synuclein, TMA106B, anything like that?
Perhaps I can start.
Great.
So in rare cases, we actually see TDP43 even. And perhaps if we get more sensitive antibodies, for example, method to see, even in the biopsy, we may see it a bit more frequently. But there's a fact that, of course, we don't like the tau. It's very, very late in the disease course than we can see it in cortex. On the other hand, we are very definite then. But I think when we combine this with the CSF, for example, I think we can get better. And then, of course, we can do like PET for even these patients. So I think this is case for that. And I think I'm very eagerly waiting more about the results about the CSF cells, because I think that reflects something very novel.
Yeah, I think, it's right. We started the CSF pellet collection with Ville in 2020, and so the results I presented are for the first 2 years of that collection. And we have 4 patients that are tau only, so we're basically not really powered to do very much kind of differentially there. But I think, again, we've continued this collection now over the years since then. And I think, again, hoping that more and more biobanks adopt protocols to save the pellets, it'll be really interesting to go into FDD cohorts, to go into ALS cohorts, MS cohort. MS is probably actually the easiest one to study because you have an infiltration of cells in MS, and so that's where the majority of the CSF studies are. But other neurodegenerative diseases where we're biobanking this other tissue and patients are coming in for CSF or blood collection, I think could be really valuable to power our analyses and learn more about these cells.
All right, so I just wanted to say thank to all of you. I think it's really impressive, all the collaborative effort that you guys are building into this exciting cohort. And as you said, there are a lot of things coming up, so stay tuned for all the exciting results that this amazing team has to show you. And thank you very much, all of you, for attending, and enjoy the rest of your day.
Thank you.